All right, so today we're going over action potentials. I'm sure that everyone is going to love this lecture and that this is going to be everyone's favorite section in biopsych. But just in case you're one of the few people, which is probably most of you, that do not like biophysics and have not heard or seen a lot of this information before, just know that I also had no idea what the heck was going on when I first heard about action potentials. So if at the end of this lecture you have no idea what's going on, or if in the middle of it you start falling asleep, or you start realizing that you don't understand what I'm talking about, which is very likely for some percentage of you, just go ahead and go back to the beginning and try to not move on to the next part of what I'm talking about until you kind of grasp what it is that's going on, because action potentials can be extremely complicated. I think that they are very fundamental and important to understanding how our brain works. So I'm not going to go easy on this portion in the exam. And it's not because I'm trying to be mean or because I'm trying to make exams like harder than they should be at this level. It's just that action potentials are fundamental and crucial to neuroscience, to neurophysiology, and to how we understand the world to be. Okay, so please take your time with this lecture. Hopefully I will break it down in a way that is easily digestible, and hopefully all of this bluster about talking about how challenging it is really actually just me overthinking it and it actually not being that complicated, because sometimes that's the case. Sometimes I make things more complicated than they actually are, and people are like, oh yeah, that was actually not so bad. So all right, so action potentials. Let's give some context first about what it is that we're even talking about, or why we're talking about this. I'm pretty sure I've shown this slide already at some point. But, we're going back to the context of the nervous system and what the nervous system is doing for us in a way that we can feel and see. Because obviously when it comes to your heart pumping, right, or you breathing, these are things that we're a lot of times not going to be conscious of. Obviously we're not conscious of, some random glutamate neuron or GABA neuron, synapsing somewhere in our brain right now to allow things to happen for us to continue to survive. But one thing that we do care a lot about is our perception and our ability to see what's actually happening in the universe, right? So being able to see this stupid waterfall, being able to taste the sweet, popsicle, being able to smell a sunflower. I don't know if sunflowers have very much of a smell or not, but there we go. So these activities are releasing chemicals or some kind of stimulus out in the universe. In this case, it's photons. In this case, it's sweet sugar molecules. In this case, it's odorants. And that information is communicating with nerves. Those nerves are then sending signals. We have our little chain of events that we talked about, I think, in one of the second lecture or whatever, where we're getting agonization of some activity. It's what agonization is referring to, of some receptor that is picking up information, some activity of some receptor, some receptor that's picking up information about the outside world. It's then sending that information down, or up to our brain, and then we are able to perceive and have all that happen. So, but what is actually happening here? I mean, these little arrows are showing the flow of information, but what is the flow of information, and how do neurons communicate? In general, if someone says a neuron is activated, they are referring to the idea that the neuron has fired an action potential. Okay, an action potential is the flow. Let's just give you your actual definition before I start to elaborate. But action potential is a change in voltage across the cell membrane that results in a subsequent propagation of this electrical signal down the length of the neuron. Okay, so we're going to get more into chemical communication of the nervous system, because that's primarily how neurons communicate to each other. But in order to release that chemical information, there needs to be a signal to release it. And that signal is the discharge of electrical activity. So again, an action potential is a change in voltage across the cell membrane that results in a propagation of this electrical signal down the length of the neuron. So the neuron is receiving information here up at the dendrites from presynaptic cells, and it is getting an influence of ions that are then going to cause a discharge of electrical activity. Okay? So this is the primary and fundamental way that neurons are functioning. Now, when you're talking to electrophysiologists or people that are measuring the actual electrical properties of neurons, they have very specific jargon about what, and by jargon I just mean scientific vocabulary, that they're going to be referring to when they're talking about a neuron being fired or activated or being on. It gets very complicated, the nuance in the way that scientists are referring to various, properties that they're measuring. But in general, like I say here, if someone's saying a neuron is activated, they're talking about that neuron firing an action potential. If they're talking about inhibition, they're saying that neuron is less likely or is being stopped from firing an action potential. Okay, so like I said in the last lecture, we're going to have to talk a bit more about the cell membrane. We have a phospholipid bilayer here. This is composed of phospholipids. One of the components of this that is very important is the hydrophilicity, or the fact that the heads are hydrophilic, meaning that they're attracted to water, and that the tails are hydrophobic, meaning that they repel water. And these characteristics is what gives cells their shape and gives neurons their shape. It also is what causes a separation of charge or a differential distribution of ionic concentration. So what the heck does that even mean? Well, I'm not going to assume that people know what ions are. I didn't even know what an ion was probably until fairly recently. I'm kind of joking, but I'm just saying that, if you haven't had a physics class in a long time, or maybe you never had a physics class, which was my situation somehow when I got to college. I'd never taken physics or chemistry, so a lot of this was really confusing initially. But anyways, we have cations and anions. Cations are positively charged ions. Anions are negatively charged ions. Okay, so we're not going to go into what positive and negative charge is, but just know that cations are positively charged ions, anions are negatively charged, and the makeup or which ions are present in the extracellular or intracellular space is ultimately what's going to cause the neuron to have a particular charge. In the intracellular space, we have a different ionic concentration than the outside of the space. And these ions can't just flow across the membrane without some method of transportation. So they can't just go through this phospholipid bilayer. So since that is the case, since the ions can't just flow wherever they want to go, and we're going to talk about molecular or ionic movements later on, but basically that causes what's called a separation of charge. So there is some kind of electrical potential between these ions wanting to communicate or wanting to move around, and since they can't freely move, that's what's going to give us the basis for this electrical activity that we're going to be talking about. So we need to know that, again, cations are like sodium and calcium and potassium, and negatively charged ions are like chloride. So we have more sodium and calcium outside the cell than inside the cell, and inside the cell we have more potassium. Now that is a positively charged ion, and so you might think, okay, well, we have a lot of this positively charged ion, so maybe it's the case that the inside of the cell is positive. But actually, the inside of the neuron is negative relative to the outside. And why that is the case is because there are these large negatively charged proteins that change the ultimate charge on the inside of the cell relative to the outside of the cell. Now, I'm going to keep on saying that, and that's very important to understand that when I'm saying negative charge or positive charge, it's relative to some other measurement, right? So it doesn't really make any sense to say it's just negatively charged with no context. What that means is that if you're comparing the two spaces, the intracellular and the extracellular space, then what that's going to result in is a difference in charge. Either the intracellular or the extracellular is going to have to be more positive or less negative relative to the other one until we get to a depolarization event. So, hold on. Okay. So, so on the outside of the cell, also one thing to note is that, now this is not, I'm not going to like grill you on this on an exam, but just in case people are interested in this, the actual neuron itself, if you were to look at the entirety of the neuron, that's not actually going to be more negative than the outside of the cell. It's just at the cellular membrane. So it's not the entirety of the neuron. It's just what we're talking about when we're saying that neurons are more negative than the outside of the cell. We're talking about at the level of the cellular membrane, just at like in between these two spaces here, okay? So just a little fun fact in case people go further into neuroscience or into neurophysiology and discover that later on, you're not surprised. But anyway, so yeah, on the outside of the cell, we have more sodium and more calcium. On the inside of the cell, we have more potassium, but it's still negatively charged relative to the outside of the cell because of the fact that we have these big negatively charged proteins. Okay, so we're going to have to take a look here at what's happening with ions. Okay, so like I said, we have a lot of sodium and calcium and chloride on the outside of the cell. We have a lot of potassium and negatively charged proteins on the inside of the cell, and they are going to congregate by the membrane because ions are going to be wanting to obey. They're not sentient, so I'm talking about them, colloquially like this, but they have properties that cause them to go places, okay? And depending on whether we're talking about positive or negative ions, they're going to want to move in certain ways, okay? So I know I'm being very nebulous here, but I'm just trying to give some context before we start getting technical. So they are attracted to this membrane because of their negative or positive characteristics. So in order to understand why ions are moving in a certain way, is that we have to understand ionic movement. We have to understand the passive mechanisms that cause ions to move in one way or another. So these two mechanisms that we're going to talk about are diffusion and electrostatic force or pressure. It's the same thing. Whether we say electrostatic force or we say electrostatic pressure, it's the same thing. So the first concept is pretty easy to understand, which is diffusion. So let's say that I take all of my molecules, I have not defined, my ions, I haven't defined them, and I'm going to push them all to a corner, and I'm going to keep them there with this barrier. They're all going to be concentrated on this, on this side of the screen here. Let's say that I remove that barrier. Diffusion is this idea that these ions don't want to be all clumped up, and so they're going to disperse until they're filling up the rest of the area that they can. So it's the same thing when you're talking about like water molecules. When you pour your water into a bowl, it's going to diffuse and it's going to fill the bowl and it's going to find its level. It's a similar thing here with ionic concentration. When you have a particular ion, let's say sodium, and you put all these sodium together, they are going to repel each other and want to diffuse away from each other when given the opportunity to do so. So this is diffusion. Ionic diffusion will be on the exam for sure, 100%. So you must know what diffusion is. The other force that we'll talk about is electrostatic force or electrostatic pressure. And this is something that, I'm pretty confident that we're all familiar with, which is that opposites attract. So if you have a sodium ion and you have a chloride ion, those are going to want to get closer together, right? So if you have a broad concentration or a broad charge, let's say like the outside of the cell, which is primarily going to be positive relative to the inside of the cell, a positive ion is going to want to get away from all that positivity. I'm saying positivity like it's I'm sorry. In any case, what I'm saying is that opposites attract. So, positive ions and negative ions are going to be wanting to get close together. Ions of the same concentration or the same charge are going to repel. So that's this one right here. So we have opposites attract, and the same are going to repel each other, just like on the, like when you were playing with magnets as little kids. Okay, so keeping in mind our characteristics of our ions and how ions want to move, they want to go away from higher concentrations of the same ion, and they also want to get away from areas that have a particular charge based on their own charge. So sodium is going to want to get away from areas of high concentrations of sodium, but also areas of high positive charge. But what would happen if we broke a hole in our phospholipid bilayer? I already told everybody that we have a different ionic makeup on the outside of the cell relative to the inside of the cell. So if we broke a hole in our cell membrane, what would happen to our ions? Well, we have to first take into account the concentration of each ion, or how much sodium is out here. In this case, since there's a lot of sodium, the sodium would want to flow away from the other sodium into where there's a lower concentration of sodium. So in this case, the sodium ions would rush into the cell. And it's doing that for the first reason of diffusion. Okay, so we first talked about diffusion, and then we talked about electrostatic force, right? So the first reason why the sodium is going to want to get into the cell is because there's so much sodium out here. But another thing that could happen is the potassium would want to get out of the cell. Okay, so let's say that this hole was permeable to both sodium and potassium. Sodium would want to go in, but potassium would want to go out. Okay, so these are two potential outcomes of opening a hole in our phospholipid bilayer. Now, of course, if you actually just ripped a hole in the phospholipid bilayer, your cell would die. So that's not what happens. What we actually have are proteins called ionophores, which are permeable to certain ions. We have sodium channels, we have potassium channels. You do need to know that for sure, that we have sodium channels, we have potassium channels. These are called ionophores. One thing you do not need to know, but I'm just pointing out that, in other places like the kidney, you have aquaphores, which are, again, proteins that allow the passage of, water. In any case, if you were to put a potassium-sensitive ionophore or a sodium-sensitive ionophore, that would determine what happens to these ions. One that is sensitive to potassium would allow the efflux, right? Efflux means, the potassium running away, essentially. If you put a sodium-sensitive ionophore, then that would allow the sodium to go into the cell. And this is based on the concentration gradient. But we also have to take into account the charge that these ions find themselves in. So sodium is a positively charged ion, which means that it's going to want to get away from levels of high positive charge. So if you were to open up a sodium-sensitive ionophore, then what would happen is the sodium would want to go down its electrical gradients away from this high level of charge and would flow into the cell. Now, this influx of positive ions would do something kind of interesting, right? So the inside of the cell, I already said, is more negative. But if we allowed the influx of a bunch of positive ions, this would result in the intracellular space becoming more positive. What if simultaneously to this, we opened up another protein, another ionophore, that was sensitive to potassium? Well, potassium, although there is a high concentration of it in the intracellular space, the electrical valence change on the inside of the cell would force the potassium out. Okay, because again, ions are constantly, going back and forth from this balancing act between its chemical and its electrical gradient. So it's going to want to move away from higher levels of itself, but it also needs to move away from, its relative charge state. So if we allowed the influx of a bunch of sodium ions, this would cause the inside of the cell to become more positive, which would then force the potassium to move out of the cell. So how do ions flow across the membrane? Well, I already spoiled this for you moments ago. I apologize. But they do it through ionophores. So these ionophores can be just there all the time and allow the influx or efflux of certain ions. But some of them are there and they are like doors that open when certain characteristics are met. So in this case, we're at 20 minutes, so we're doing good. We're going to be talking about voltage-gated ion channels. Now, a voltage-gated ion channel is a protein that spans the phospholipid bilayer. So here we have, again, our phospholipids, and then we have this protein here that is on both sides of the phospholipid bilayer. And depending on the voltage that's present at the membrane, this protein will be in a particular conformational state. Okay, so if you haven't had any, this could be your first biology class. If that's the case, then we're talking about proteins, which are just large chemicals. Okay, and they have a particular shape. Okay, so let's say that at rest, this voltage-gated ion channel is just connected like this. Now, let's say that we get an action potential, which is a, electrical flow down the neuron. This voltage-gated ion channel, as the name suggests, is going to react to that change in voltage, causing it to open. This is called a conformational change, okay? So, change in the membrane potential causes this voltage-gated ion channel to open, which would then allow the influx or the efflux of the various ions. Okay. But what causes this ionic distribution? Okay, why are there more potassium channels on the inside than the outside? And why are there more sodium on the outside than the inside? I know that you guys are like dying to know this. So the reason that we have this particular ionic concentration is because of active transport mechanisms. So here is another graphic trying to show us this phospholipid bilayer. We can see that we have our phospholipids, and we can see that on the inside of the cell we have a negative charge. On the outside of the cell we have a positive charge. And we can actually see from this picture here that potassium is flowing relatively, freely outside of the membrane. And that's because the membrane has ionophores that potassium can just kind of go through. But what happens is we have something called the sodium-potassium pump. Now, like action potentials, the sodium-potassium pump is one of those things that is just super fundamental to neuroscience. So you absolutely are going to have to know about the sodium-potassium pump. You will need to know that the sodium-potassium pump ejects 3 sodium ions from the inside of the cell to the outside of the cell. And it takes in 2 potassium ions. Okay, so the fact that it's shooting out 3 sodium ions, but bringing in only 2 potassium ions is going to help explain why there's more sodium on the outside of the cell relative to the inside of the cell. So what is the sodium-potassium pump? It is a protein that spans the phospholipid bilayer, and it ejects 3 sodium ions, and it brings in 2 potassium ions. Now, a more kind of technical name for this is a sodium-potassium ATPase. I'm not going to be asking you about this kind of thing, but in case you're interested, this energy, excuse me, the energy for this process is ATP. Okay, so you have an ATP molecule and H2O that's coming up to this sodium potassium pump. In order to fuel this, you have to remove a phosphate group, and you get adenosine diphosphate instead of adenosine triphosphate. So you have 3 phosphate groups. It comes up, it cleaves one of those phosphate groups and shoots out adenosine diphosphate because now it's removed 1 phosphate. And that is the energy that allowed this protein to eject the 3 sodium and bring in the 2 potassium. So ATPase is just another name for the sodium potassium pump. This is from a neurobiology textbook, so they're not saying pump, but it's the same thing. So, and this is a example of active transport. So previously I talked about passive transport, which is the ability of the ions just to move based on their particular ionic characteristics. But this mechanism, it's called active transport because it's actually forcing ions to move against their will. Not that they have a will, but you know what I mean, like against their characteristics. So it's like it'll force the ions to go against their concentration gradient. Because remember, the sodium doesn't want to go outside, but this active transport mechanism, this sodium potassium pump, is saying you do have to go inside. So just to be clear, what you need to know about this is, 1, that active transport moves ions against their concentration gradients and against their electrical gradients, okay? Number 2 is that this is a sodium potassium pump. It is ejecting 3 sodium ions and bringing in 2 potassium ions. 3, is that 3 or 4? 3, you need to know that this is working at all times. It's always happening. It never stops. And I think that's all you need to know about the sodium potassium pump for this purpose. Also, this pump is what brings the neuron back to its baseline, to its homeostatic or its baseline charge. Okay, so in this slide, we're going to go ahead and put everything together that we've gone over so far. We have the phospholipid bilayer here, and we have the extracellular space being more positive and the intracellular space being more negative at rest. And in this state, it is being referred to as polarized. Polarization just meaning 2 ends of the pole. So if you're talking about Donald Trump as a polarizing figure, or the stance, the United States stance in Gaza is polarizing or whatever, we're talking about opinions being on opposite ends of a spectrum. In this case, we're talking about a neuron being polarized because the extracellular side of the membrane is positive and the intracellular side is negative. When an action potential occurs, we refer to that as depolarization. And that's because the intracellular space is now positive, meaning that it is now similar on both the extra and intracellular sides of the membrane. So how does this occur, and what is the action potential actually talking about in terms of all the various steps that are occurring? So let's go ahead and look at our little graph here and orient ourselves to the axes. On the x-axis, we have time, and this is also just the various stages of the action potential. On the y-axis, we have the membrane potential measured in millivolts. Now, the resting potential, or when a neuron is at rest, has various different numbers that you may see depending on the textbooks that you read. Generally, it's between -65 and 70. This book is looking at saying that it's at -70 millivolts. That's fine. The point, though, is just that a neuron at rest has a particular voltage, right? And we also need to talk about a threshold for an action potential. So once the membrane voltage reaches a certain millivolt, we're going to have an action potential. This is called the all or nothing principle. It is something that you need to know for sure. So once the threshold is reached, there's nothing you can do. An action potential is going to propagate. In this graph, it's saying that that threshold is -50 millivolts. So yeah, I think that that's basically all you need to know about the graph. And we are going to now go over each step. So the first one is looking at the resting potential. I alluded to this previously, but there are basically potassium ionophores that are just open that allow passive diffusion of potassium across the membrane. Now, as we already went over though, there is always going to be a higher concentration of potassium on the inside of the neuron, and that's because of the sodium-potassium pump constantly bringing potassium into the neuron against its concentration gradient. There's also closed sodium channels that are voltage gated. And there's also voltage gated potassium channels. So we have passive ionophores for potassium, which mean that it's just always open and potassium can just do whatever it wants with this ionophore. And then we have voltage gated ionophores, which we've already gone over, but I'm going to say it again. We have voltage gated sodium channels and we have voltage gated potassium channels. So you need to know that both of those things exist. Now, what's going to happen is that once the neuron starts to get information from the previous cell, so we're getting the opening of sodium channels due to some kind of chemical communication that we'll go over in the next video, I think, for synaptic transmission, we're going to have an increase in the membrane voltage. Now, once the membrane voltage reaches negative 50 millivolts, or the threshold, then we're going to have the opening of all of the membrane or the voltage gated sodium channels. Once that occurs, we're going to have a mass influx of sodium. So why does that occur? Well, it happens, why does the sodium go in? It's happening because the sodium is traveling down its electrical chemical gradient, meaning that's going away from all of the extra sodium that's in the extracellular space, but it's also going away from this increased positive state. Once this happens, we're going to have a huge increase in membrane voltage. So now the neuron is in a depolarized state, okay? So both the extracellular and the intracellular space are both positive, therefore there's no more polarization. The neuron is depolarized. So if you hear depolarization, you know that means action potential has occurred. At the top of this phase of, phase 3, potassium channels will open. Voltage-gated potassium channels that were otherwise closed. Sorry. Okay. Once these voltage-gated potassium channels open, we're going to have a mass efflux of potassium. And why are we having that mass efflux of potassium? It's because of all of the sodium that's now gone into the neuron and caused the intracellular space to become positive. So now the potassium is trying to travel away from its electrical gradient, right? So it's trying to travel down and away from this positive environment. This mass efflux of potassium causes a decrease in the membrane voltage and hyperpolarization. So it's not written out on the slides, but I said that you need to know the all or nothing principle, which means that once you reach this threshold, you're going to have this action potential no matter what. Like once it's like firing a gun, once you pull the trigger all the way, you can pull the trigger and get right up to the point, but once you press it, the gun is firing. Or like with the toilet, as soon as you can start, allowing the water to flow, but once you reach a certain level, the toilet will flush. Same thing here. The all or nothing principle is saying once we reach the threshold and those sodium channels are open, we're getting an action potential. Similarly here, what is not listed but you need to know is that during this hyperpolarization event, getting another action potential is extremely difficult. It is possible to get an action potential before the voltage has gone back to its resting potential, but it is very difficult and it causes a lot of stimulation and much greater excitation than it initially required. And at some point, you actually can cause the neurons to stop firing altogether when too many action potentials have occurred. So What also happens here at the top of this, the top of this action potential is that the sodium channels are inactivated. So we can see that here. So the sodium channels are closed and the sodium can no longer flow through because the ionophores have again changed their conformation, resulting in the passageway being closed. The red line here is indicating that after some period of time, it gets back to its resting phase and all of the voltage gated channels are closed, and you have a normal or a depolarized state again. A polarized state again, and that's because of that sodium potassium pump essentially bringing everything back to normal. So that is the action potential in a nutshell. It is kind of complicated, and you may need to go through these steps again in order to fully get it. So, questions you can ask yourself to see if you understand the concepts or like what's happening here with the action potential are, for instance, right here, when we reach the threshold and this voltage gated sodium channels are open, why do we get an increase in the millivolts here? Why does this happen? I'm going to give you a second to see if you can figure that out because this is extremely important. The reason why the membrane potential has increased is because there is a mass influx of sodium. Sodium is a positive ion, so if we put more, if we increase the amount of the positive ion on the inside of the cell, it's going to cause the intracellular space to become more positive, right? Another question to test yourself here. Why does opening these voltage-gated sodium channels result in sodium going into the cell, right? So it's like, this not active transport, it's passive transport, right? So why? I mean, there's no protein forcing the sodium in. The sodium is just, for some reason, flowing into these open sodium channels. So go ahead and see if you can figure that out. The reason why is because of the concentration and the chemical, the electrical gradient that's on the outside of the cell. Sodium is a positively charged ion, so it's going to want to flow into the negatively charged space because of the opposites attract and the similar repel characteristic of ions. And then also it's because of the chemical gradient outside of the cell. There's so much sodium outside of the cell already that the sodium is going to want to get away from that high concentration. So that's the diffusion port, right? Let's see here. What's another one that we can talk about? Okay. Why is it the case that although there are these open potassium channels all the time, so potassium can just flow in and out whenever it wants, why is there still more potassium on the inside of the cell? Well, that's because of the sodium-potassium pump. So the more potassium, the active transport mechanism, meaning that the protein that's forcing potassium against its electrochemical gradients, is always working. So even though we have this ability for potassium to flow in and out, we always have more potassium coming in the cell because of that sodium-potassium pump always working. Let's see here. You may want to ask yourself, what is the all or nothing principle? So if you don't know what that is, then rewind the video because I just described that. Can another action potential occur once the neuron has gotten to this hyperpolarized state? The answer is yes, but it requires a much higher level of excitation, and you can actually get to a point where the neuron can no longer fire after repeated stimulation. So, a question could be, is it possible for a neuron to not be able to fire an action potential? And the answer to that would be yes. After repeated firing, an action potential can no longer occur. I don't trick anybody on the exams, but a potential thing that could seem like a trick would be, can an action potential fire after hyperpolarization? Number one, if that sounds like total jargon that you don't understand, then again, you're going to have to rewatch this portion, but the answer to that would be yes, it can, but it would require a high level of excitation. Okay, so I think that's it for this figure. And then, of course, we have all of our potential questions that may appear on the exam. Also, I put this here. It says, what are the two passive mechanisms for ion movement? And I put, this is not a trick. The answer is directly above, and that is true. The two passive mechanisms of ion movement is molecular diffusion and electrostatic force. And I just put that there because sometimes people overthink things, and I might ask something very basic like, what is a cation? Or maybe the question in the exam says, maybe there's a question right above that says sodium is a cation, and then the next question says what is a cation, and then sodium is one of the answers. Like if that is the case, it is not a trick. I do not want to trick anybody on purpose, and there are no trick questions. And if there are any, it's completely on accident. So this is never going to be the case where I'm like, what are the two passive mechanisms for ion movement? And, I don't even know how that could be a trick, honestly. But yeah, I'm not structuring the exam or the questions of the exam in a way to try to confuse you. The answers to the questions are going to be based on the facts that were presented in the PowerPoint. So, obviously, how many sodium ions does the pump kick out? How many potassium ions does it bring in? It kicks out 3 sodium ions, it brings in 2 potassium ions. So, that kind of thing. What is the resting membrane potential? It's, 70 millivolts. You know what I mean? So that might be, again, one of those things where you might be confused about, like, what is it that I'm actually asking? Hopefully, the answers, it's all going to be multiple choice. So that's not trying to be philosophically confusing, although I now realize that it might be. What does it mean when someone says the neuron was depolarized? Again, this is not short answer on the exams because that could technically have multiple answers, but generally speaking, it means that the neuron is fired in action potential. It could also mean that the voltage of the neuron is now the same on the outside and the inside of the membrane, but the potential answers will be clear. So again, this is a particularly heavy section of the class, but if anyone has any questions about this, feel free to reach out. I understand that it can be challenging for people, but we will see you in the next lecture, and thankfully, the next couple lectures I think are on neurotransmission, which can also be a little bit confusing. And then we have anatomy. And then once we're out of kind of the physiology anatomy section, then we're going to get into things that might be a little bit less confusing. But yeah, see you in the next one.